One of the questions that we have is how effective would chemostat treatment be if the virus were to mutate? Since we know that SARS-CoV-2 and SARS-CoV-1 are rapidly mutating viruses, and viruses in general are known to develop drug resistance, we were wondering what would happen to viral fitness and pathogenicity if, for example, its S protein mutated. So it's possible that the spike protein could mutate to no longer need TMP RSS2, the serine protease, or other serine proteases from the host in general. And this would mean that chemostat treatment, which inhibits or serine proteases, would no longer be effective. Of the experiment that we're proposing is to investigate what would happen to viral fitness and pathogenesis in the event that the SARS-2 S protein mutated and became resistant to and no longer needed serine protease-based priming. So the virus would become resistant to chemostat treatment. What we would do in this experiment we're proposing is we would use a human lung cell line like Calu-3 that we've used before, and we would treat it with really low concentrations of chemostat, and then start inoculating it with wild type SARS-CoV-2. And of course, due to concerns of safety, we would do this in a BSL-4 facility. Next, we would continue to passage these cells and propagate the virus, but every subsequent passage, we would increase the chemostat concentration slowly. And so we would slowly increase the chemostat concentration from a proposed value of 0.2 micromolar to eventually 10 micromolar over a series of many passages where we continue to propagate the virus. We chose 10 micromolar because this is one of the doses that the virus was treated. Next, what we would do to monitor the virus is just as it was done in figure 4D, we would continue to look for genome equivalents per ml using RT-qPCR to quantify the amount of viral RNA. And what we would hope to see is that with increasing passaging, while concentrations are increasing, the amount of GE per ml would continue to increase, meaning the virus is replicating over time as the chemostat concentration is increasing. We would hope to see in the experiment that with increasing chemostat concentration, the amount of viral RNA would increase. The way we would monitor the virus is by doing what was done in figure 4D that was covered earlier. We would monitor the genome equivalent per ml using RT-qPCR, which quantifies viral RNA. And we would hope to see increasing concentrations of GE per ml or viral RNA as we continue to passage cells and propagate the virus, while chemostat concentrations are also increasing. Eventually, we would end up seeing really high GE per ml values or really large amounts of viral RNA, even with a high concentration of chemostat treatment. This would indicate to us that SARS-CoV-2 had mutated to infect the host cell without needing priming by the serine protease TMPRSS2. Next, we would isolate the S protein and compare its amino acid sequence to wild type SARS-CoV-2 with the hope that maybe we could identify novel domains or we could see what had changed about the protein structure. It's possible that the amino acid sequence could change so that there's no longer a serine in place where it used to get cleaved to become an S1 and S2 subunit. It's also possible that the virus ends up not needing priming at all. Next, we would use pseudotyped VSV and transfect it using a retroviral vector with this new mutated S protein that we hope to see. We'd want to use a pseudotyped VSV in this case because we want to make sure any potential differences we see in pseudotype entry are due to mutations in the S protein, which we would give to the pseudotype exclusively, and not due to some other mutation that the virus ended up accruing, and not due to some other mutation the SARS-CoV-2 virus ended up accruing while we were mutating it with camostat treatment. What we would do is use that same luciferase assay to determine pseudotype entry into human lung cell lines. The next thing we'd want to do in this case is determine what this mutation has done on the ability of pseudotype VSV with this mutated S protein to enter host cell lines. We would then maybe
Perhaps we
even
would
treat
see
these
a
cells
shift in
with
dependence in the S
camostat or
protein from
E46B,
needing a serine
cysteine
protease,
protease
like
inhibitor,
TMP,
to see what
to
would
maybe
happen.
needing a cysteine
Perhaps
protease.
the new S
A
protein
study like this
allows the
would help us
virus to
determine
use
what the
another
effect of
protease.
mutations
Perhaps
that
it
confer resistance
shifts the
to serine
dependence
inhibitors would mean for viral pathogenesis and viral spread. This would be critical in determining how effective this potential treatment option is in humans.